For centuries, Afghanistan has been described as a place conquered by everyone and owned by no single ancestry. Persians marched through it. Greeks founded cities there. Steppe herders moved across its northern approaches. Buddhist kingdoms rose and vanished. Arab armies brought Islam. Turkic dynasties ruled its valleys. Mongol forces crossed the mountains. Later empires pulled the region toward Iran, India, and Central Asia at the same time. That history created one of the most persistent questions in Asian population genetics. If so many peoples passed through Afghanistan, who are Afghans genetically? Were the Pashtuns descendants of ancient Israelites, as some traditions claimed? Did Alexander's soldiers leave a detectable Greek signature? Are the Hazaras mainly descendants of Mongol armies? Are Tajiks simply Eastern Persians? And did all these groups begin as separate populations, or did their differences appear later? When scientists finally compared Afghan genomes, paternal lineages, maternal lineages, and ancient DNA from the wider region, the answer was not one origin. It was a layered genetic crossroads built over thousands of years with one shared foundation and several later histories stacked on top of it. That is why the title of this story needs one important correction. There was never a single Afghan DNA mystery waiting for one perfect ancient population to match it. Afghanistan is home to Pashtuns, Tajiks, Hazaras, Uzbeks, Turkmen, Nuristanis, Balakh, Aimak communities, and other groups whose histories are not identical. Modern national borders also arrived much later than most of the migrations that shaped them. A person living in eastern Afghanistan may share deep ancestry with people across the border in Pakistan. A Tajik from northern Afghanistan may connect genetically with Iranian-speaking populations across Central Asia. A Hazara genome may preserve a stronger East Asian signal than a neighboring Pashtun genome. Yet beneath those differences, several studies found evidence of ancestry shared across Afghan populations that reaches back before most modern ethnic identities existed. The mystery was not which civilization created the Afghans. The real mystery was how a common regional foundation became divided into the genetic patterns visible today. One of the first large attempts to answer that question examined Y chromosomes from major Afghan ethnic groups. The Y chromosome follows the paternal line, so it does not describe a person's entire ancestry, but it can reveal how male lineages expanded, split, and survived. Researchers analyzed Pashtuns, Tajiks, Hazaras, and Uzbeks and compared them with thousands of men from surrounding regions. The result immediately destroyed the idea of one uniform Afghan paternal profile. Some lineages were common in Pashtuns and Tajiks, but rare in Hazaras and Uzbeks. Others showed almost the opposite pattern. The most frequent lineages included branches of R1a, C, J2, and L, each associated with different parts of Eurasian population history. But the researchers also found something more important than any individual haplogroup. Their models suggested that the major Afghan groups still shared ancestry from an older regional population, and that strong differentiation between them began much later, around the Bronze Age. That Bronze Age timing matters because it shifts the story away from the famous conquerors most people know. Alexander arrived only in the 4th century BCE. The Arab conquests came roughly 1,000 years later. The Mongol expansion came more than 1,000 years after that. But the genetic separation between some Afghan populations appears to have begun thousands of years before the Mongols and long before Islam, during the period when farming communities, oasis settlements, pastoralist networks, and early urban civilizations were transforming Central and South Asia. In one study, the estimated beginning of major paternal structure among Afghan groups centered roughly around 4,700 years ago, although the confidence interval was broad. That does not mean modern Pashtun, Tajik, Hazara, or Uzbek identities existed 4,700 years ago. They did not. It means the ancestral populations that later contributed to those identities were already beginning to experience different movements, marriages, and geographic pressures. The geography makes that easy to understand. Afghanistan sits between the Iranian Plateau, the Central Asian steppe, the Indus world, and the mountain corridors of the Hindu Kush. Those mountains do two opposite things at once. They create barriers that can isolate communities, but they also contain passes that funnel movement through predictable routes. For ancient travelers, armies, traders, herders, and migrants, Afghanistan was not simply a destination. It was a junction. Gene flow could arrive from the west through Iran, from the north through Central Asia, from the southeast through the Indus region, and later from farther east through Turkic and Mongol expansions. The genetic landscape therefore behaves less like a tree with one trunk and more like a braided river, where streams separate, reconnect, and carry different sediments downstream. A second major study expanded the picture by analyzing more than 500 Afghan individuals from Pashtun, Tajik, Hazara, Uzbek, and Turkmen populations using mitochondrial DNA, Y chromosomes, and autosomal markers. Autosomal DNA is especially useful because it represents ancestry inherited from all recent family lines, rather than only one paternal or maternal branch. The researchers described Afghanistan as a mosaic of Eurasian components, rather than a population that formed in complete isolation. Their conclusion was striking. The Hindu Kush appeared to be a zone where gene flows from different parts of Eurasia converged. Afghanistan was not genetically simple enough to be described as purely Central Asian, purely South Asian, or purely Iranian. It carried signatures related to all of those worlds in different proportions depending on the population. This is where the Pashtun story becomes especially interesting. Pashtuns are one of the largest populations spanning Afghanistan and Pakistan, and their origin traditions have attracted extraordinary speculation. Some clans have preserved stories connecting themselves to ancient Israelites. Other theories linked Pashtuns to Greeks left behind after Alexander the Great's campaigns. Still others emphasized Iranian or Indo-Iranian origins based on the Pashto language, which belongs to the Eastern Iranian branch of Indo-European. Genetics has tested parts of these stories, and the results are far less dramatic than the legends. Studies of Afghan Pashtun Y chromosomes found high frequencies of R1A lineages and other paternal branches also present across parts of South and Central Asia. Researchers did not find the specific Balkan-associated Y chromosome signal that would support a simple story of large-scale descent from Alexander's Greek soldiers. That does not prove that no Greek individual ever contributed ancestry to any Pashtun family. After more than 2,000 years, small contributions can disappear, spread widely, or become impossible to identify as specifically Greek. Alexander's armies also included people from many regions, not one genetically uniform Greek population. What the data does reject is the stronger claim that Pashtuns as a whole can be explained as descendants of Macedonian or Greek settlers. Their dominant paternal ancestry fits much better within the broader genetic landscape of South Central Asia and the Iranian-speaking world. Pashtun samples from Afghanistan also show strong affinity with Pashtuns across the modern border in Pakistan, reminding us that political borders can divide a population whose genetic history is much older than the state line. The Israelite claim is even harder to test. Some Pashtun traditions connect tribal genealogies to the lost tribes of Israel, and that story has been repeated for generations. But genetics cannot confirm an ancient identity merely because a modern population shares a haplogroup with Jewish populations. Common Y chromosome branches can be thousands or tens of thousands of years older than Judaism itself, and can appear across huge regions. The Afghan studies did not reveal a clear population-wide paternal pattern, uniquely tying Pashtuns to ancient Israelites. That does not evaluate the cultural meaning of the tradition. It simply means the genome has not provided the kind of specific evidence required to turn the legend into a demonstrated population origin. Instead, Pashtun genetics points toward something broader. Their ancestry reflects the long interaction zone between the Iranian Plateau, the Indus region, and Central Asia. In paternal datasets, Pashtuns and Tajiks often share relatively high frequencies of R1A compared with Hazaras and Uzbeks. They also carry lineages such as L and other branches common in parts of the Indian subcontinent. One study found that Pashtun and Tajik paternal profiles showed notable affinity with northern and western Indian populations, while also connecting toward Iran and western Eurasia. This is exactly what geography predicts. The Hindu Kush did not separate South Asia and Central Asia completely. It connected them through selected corridors, and Afghan populations preserved evidence of those connections. Tajiks tell another version of the same crossroads story. Afghan Tajiks speak Dari, a variety of Persian, and culturally they are closely connected to the Persian-speaking world. Genetically, however, Persian-speaking does not mean genetically identical to modern Iranians. Studies place Afghan Tajiks within a Western Eurasian-leaning Central and South Asian continuum, with ancestry relationships extending toward Iran, Central Asia, and northern South Asia. Their paternal profiles contain substantial R1A as well as J2 and other lineages. In broader autosomal comparisons, Tajik populations tend to carry more West Eurasian-related ancestry than many Eastern Central Asian populations. A recent whole genome project across Central Asia again found Tajik groups positioned toward the West Eurasian side of the region's ancestry spectrum. But language is not a perfect genetic map. Populations can change language through political dominance, trade, religion, education, or elite influence without being replaced biologically. Afghanistan provides several examples of this. Hazaras today mainly speak Hazaragi, a Persian variety, yet their genomes contain a much stronger East Asian component than most Persian-speaking Tajiks. Uzbeks speak a Turkic language and also show substantial Eastern Eurasian paternal ancestry. But the relationship between language and genes is not one-to-one. The genetic layers often predate the languages now spoken. In some cases, a population changed language while preserving much of an older genetic profile. This is one reason Afghan history cannot be reconstructed by linguistics alone. No population illustrates the impact of later conquest more clearly than the Hazaras. Hazara traditions and physical anthropology have long connected part of their ancestry to Mongol or other Eastern Eurasian groups that entered the region during the medieval period. Unlike many famous descent legends, this one receives meaningful support from genetics. Afghan Hazara samples show high frequencies of Y chromosome lineages associated with Eastern Eurasia, including branches of haplogroup C that are much more common among Hazaras and Uzbeks than among Pashtuns and Tajiks. Mitochondrial studies also found a substantial proportion of Eastern Eurasian maternal lineages among Hazaras, meaning the Eastern contribution was not purely male. That detail complicates the popular image of a conquering army of men, leaving descendants among local women. The genetic evidence suggests Eastern Eurasian ancestry entered Hazara ancestors through both paternal and maternal lines. Medieval Mongol expansion likely contributed strongly, but Hazara equals Mongol is still too simple. Hazaras also carry older West Eurasian and Central Asian ancestry. Their population formed through mixture, followed by endogamy and historical isolation that helped preserve a distinctive profile. Modern whole genome research published in 2026 strengthens this picture by identifying long-range Siberian-related connections in Afghan Hazara groups and estimating major admixture episodes in parts of Central Asia roughly 650 to 1,000 years ago, a period overlapping the era of Mongol and post-Mongol movements. The timing is powerful because it connects genetic evidence to a historically documented transformation of Eurasia. Beginning in the 13th century, Mongol expansion created movement on a scale few earlier empires had matched. Soldiers, administrators, artisans, families, and displaced communities crossed Central Asia. Genetic mixing did not happen once in a single battle. It occurred through generations of settlement, political restructuring, marriage, migration, and the creation of new social groups. Hazaras emerged from that world, but their DNA also reminds us that medieval ancestry was added to populations already shaped by much older Bronze Age and Iron Age histories. Uzbeks in Afghanistan show another strong Eastern component, but their story is not identical to the Hazara story. Uzbek ethnogenesis involved Turkic-speaking populations and later political formations that developed across Central Asia. In Afghan paternal data, Uzbeks carried high levels of haplogroup C, as well as other Western and Eastern Eurasian lineages. Their genomes reflect the repeated mixing that characterizes Central Asia generally. Turkic languages spread across enormous distances, often through a combination of migration and language shift. The genes, therefore, do not point to one original Turkic race. Instead, they reveal a patchwork of older local populations mixed with migrants arriving from farther east and north. To understand what existed before these medieval and historical layers, scientists needed ancient DNA from the wider region. Afghanistan itself remains difficult for ancient genomic research because decades of conflict, limited archaeological access, preservation problems, and the destruction or dispersal of collections have left major gaps. But a landmark 2019 ancient DNA study sequenced more than 500 ancient individuals across Central Asia, Iran, and northern South Asia, including material from the broader region historically connected to Afghanistan. The study transformed understanding of how the populations surrounding Afghanistan formed during the Neolithic, Bronze Age, and Iron Age. One major ancestral stream came from populations related to early farmers and hunter-gatherers of the Iranian Plateau. As farming and herding expanded eastward, people carrying Iranian-related ancestry moved into Central Asian oasis regions. By the Bronze Age, the Bactria Margiana archaeological complex, often called Biamak, flourished across parts of modern Turkmenistan, Uzbekistan, Tajikistan, and northern Afghanistan. These were not primitive camps. They were organized oasis societies with monumental architecture, craft production, agriculture, and long-distance trade. Ancient DNA showed that the main Bimak population largely belonged to a southern Central Asian ancestry profile connected to Iranian-related farmers, with additional components reflecting the complicated population history of the region. But the Bimak world was not genetically closed. Individuals buried at Bimak sites sometimes carried ancestry different from the majority, proving that long-distance movement was already occurring. Around the turn of the second millennium BCE, steppe pastoralist ancestry began appearing in southern Central Asia. These northern groups descended partly from populations associated with the Eurasian steppe and carried ancestry ultimately connected to Bronze Age pastoralist expansions. Their arrival did not simply erase the oasis populations. Instead, different groups met and mixed along corridors running through Central Asia toward the Hindu Kush and northern South Asia. This Bronze Age movement is one of the most important pieces of the Afghan genetic puzzle because it helps explain why Indo-Iranian languages and steppe-related ancestry spread across the region. Ancient DNA from Central and South Asia shows that people carrying Central steppe middle to late Bronze Age ancestry moved southward after roughly 2000 BCE. By the first half of the second millennium BCE, steppe-related ancestry had reached populations in northern South Asia. Linguists have long recognized deep relationships between Indo-Iranian languages and other Indo-European branches. Genetics does not prove exactly which person spoke which language, but the timing of steppe migration provides a plausible demographic pathway for the spread of Indo-Iranian speech. Afghanistan lies directly in the middle of that route. Pashto is an Eastern Iranian language. Dari belongs to the Persian branch of Iranian languages. Several ancient Iranian languages were once spoken across Central Asia and Afghanistan, including Bactrian, Sogdian in nearby regions, and others known from inscriptions and historical records. The genetic evidence suggests that the linguistic transformation of the region was accompanied by real population movements, but not complete replacement. Steppe ancestry entered a landscape already inhabited by people with Iranian-related and South Central Asian ancestry. The populations that emerged were mixtures. That is the deeper reason the common Afghan foundation is so difficult to label. Should it be called Iranian, Central Asian, South Asian, steppe? None of those labels alone works because the ancestry predates many of the modern categories. The genetic components themselves formed through earlier mixtures. Early Iranian-related populations were not the same as modern Iranians. Bronze Age steppe pastoralists were not modern Europeans or modern Central Asians. Ancient Bimak people were not simply Tajiks, Pashtuns, or Uzbeks waiting for their present names. Every modern population is the result of additional thousands of years of movement after those ancient groups disappeared. The famous Silk Road later intensified this process, but even that phrase can mislead. There was never one Silk Road. There were networks of routes connecting China, Central Asia, India, Iran, and the Mediterranean. Afghan cities and valleys sat near branches of those networks. Bactria, centered partly in northern Afghanistan, became a major zone of exchange. Greek kingdoms appeared after Alexander's conquest. The Kushan Empire later connected parts of Central Asia and northern India. Buddhism spread through monasteries and trade routes. Iranian, Indian, Hellenistic, and Central Asian artistic traditions mixed visibly in places such as ancient Gandhara and Bactria. Genetically, however, cultural influence does not always leave a large population signal. Greek art can spread without thousands of Greek settlers producing descendants. Buddhism can transform religious life without changing most local DNA. Arabic can influence vocabulary, while Arab paternal lineages remain rare. One Afghan Y chromosome study found little evidence for a major Arabian male genetic replacement after the Islamic conquest. J1, a lineage common in parts of Arabia, appeared only rarely in the sampled Afghan groups. Islam changed Afghanistan profoundly, but conversion was largely cultural and political, rather than a mass substitution of the existing population by Arabian settlers. That distinction is essential for interpreting history through DNA. Empires can dominate the archaeological and written record while leaving surprisingly small genetic traces. Other migrations can receive little attention in chronicles but reshape genomes dramatically. The Mongol-era signal in Hazara ancestry is visible because movement and reproduction were substantial enough to leave a durable genetic mark. The Greek contribution is harder to detect as a major population-wide event. The Arab cultural impact was enormous, while the tested paternal genetic impact appears limited. Genes measure biological transmission, not cultural importance. There is another surprise inside the Afghan data. The paternal and maternal stories do not always match perfectly. Y chromosomes record only fathers, fathers, fathers. Mitochondrial DNA records only mothers, mothers, mothers. Autosomal DNA captures a much broader mixture. A conquest dominated by male migrants can create a strong Y chromosome signal but a weaker maternal one. A community that absorbs women from neighboring populations while keeping paternal clan identity can show the opposite imbalance. This is why scientists no longer try to reconstruct an entire people from one haplogroup. The Hazara example demonstrates this clearly. Eastern ancestry appears in both paternal and maternal lines, arguing for movement of more than just a tiny military elite. Pashtun lineages reveal connections across the Afghanistan-Pakistan border that match historical geography better than modern political boundaries. Tajik ancestry connects westward and northward without reducing Tajiks to one Iranian source. Afghan Uzbeks share Eastern Eurasian signals with other Central Asian populations, but also carry Western components, accumulated before and after Turkic language spread. The genome is not choosing between competing civilizations. It is preserving pieces of all the population movements that actually produce descendants. Modern whole genome sequencing is now making that picture finer. A 2026 Central Asian Genomic Project sequenced whole genomes from 166 individuals across 20 Central Asian and Afghan Hazara groups. The researchers identified strong differences in the balance of West and East Eurasian ancestry between populations. Tajik groups fell toward a more West Eurasian profile, while Hazaras carried stronger ancient Northeast Asian-related ancestry. The study also detected traces of Neanderthal-like and Denisovan-like segments, as expected in Eurasian populations. and found population-specific patterns potentially relevant to immunity, metabolism, and disease risk. These findings are scientifically important, but they should not be turned into claims that one Afghan ethnic group is more ancient, more evolved, or biologically superior to another. Every living Afghan is fully modern Homo sapiens. Differences in ancestry proportions are records of migration, not rankings of people. Haplogroups are not ethnic essences. A Pashtun with R1a is not automatically descended from one specific Bronze Age tribe. A Hazara carrying an Eastern Eurasian lineage is not genetically reducible to Genghis Khan. A Tajik with West Eurasian-related ancestry is not simply an Iranian copied eastward. Genetic categories describe statistical relationships among populations, not fixed identities inherited unchanged through history. The deeper scientific challenge is the shortage of ancient genomes directly from Afghanistan. Researchers can reconstruct much of the story using modern Afghan DNA and ancient genomes from surrounding regions, but a direct time series from Afghan archaeological sites would be far more powerful. Imagine sequencing individuals from Bronze Age Bactria inside modern Afghanistan, then comparing them with Kushan-era burials, medieval populations, and present-day communities. Scientists could watch ancestry change through time rather than infer every transition backward from living people. That dataset does not yet exist at the scale needed. Until it does, some questions remain open. How much ancestry from Bronze Age Bactrian populations survives in each modern Afghan group? Which migrations introduced specific steppe-related lineages into the Hindu Kush? How much of the Hazara Eastern ancestry came during the first Mongol expansions? And how much arrived through later Central Asian movements? When did the ancestors of modern Pashtun tribal structures become genetically distinctive? How much continuity connects ancient Iranian-speaking populations of Afghanistan with present-day Dari and Pashto speakers? These questions are answerable in principle, but they require more ancient DNA. Even the phrase Afghan ancestry needs care. Afghanistan, as a modern political identity, unites populations that were historically connected to different regional networks. Genetic research shows that ethnicity often predicts population structure better than the modern national label. Pashtuns on opposite sides of the Afghanistan-Pakistan border can be closer to each other than either is to some other Afghan ethnic groups. Hazaras carry connections toward Eastern Central Asia that differ from neighboring Tajiks. Uzbeks and Turkmen link into broader Central Asian genetic landscapes. This does not weaken Afghan national identity. It simply shows that nationality and biological ancestry operate on different timescales. And now the original mystery can finally be answered. Scientists did not discover that Afghans came from Greeks, Israelites, Mongols, Persians, Indians, or steppe nomads alone. They discovered that Afghanistan's populations formed at a continental intersection where all directions mattered. A deep regional foundation connected to early farming and Holocene populations was gradually structured during the Bronze Age. Iranian-related ancestry from the southwest, South Asian-related ancestry from the southeast, and steppe-related ancestry from the north became part of the population landscape. Later, Turkic and Mongol movements added stronger Eastern Eurasian components to some groups, especially Hazaras and Uzbeks. Historical empires then changed languages, religions, and identities without always replacing the people already living there. The result is not genetic chaos. It is structured diversity. Pashtuns and Tajiks tend to preserve more ancestry relationships toward Western Eurasia, Iran, and Northern South Asia, although they are not identical to one another. Hazaras show a distinctive combination of older Western ancestry and substantial Eastern Eurasian ancestry, consistent with medieval Central Asian and Mongol-era movements. Uzbeks carry their own mixture shaped by Turkic and Central Asian history. Turkmen show still another pattern. Beneath these differences lies evidence that parts of their ancestry descend from populations that were in the region before the modern ethnic names formed. That may be the most important result because it reverses the popular image of Afghanistan. History books often describe the country as a graveyard or highway of foreign invaders, as though its people were simply the leftovers of everyone who passed through. Genetics shows something different. Incoming groups repeatedly encountered populations that were already there. New ancestry was added, sometimes dramatically, but older ancestry survived. Conquerors became absorbed, languages changed, religions changed, tribal names changed, political borders moved. The underlying population was transformed without being erased. That survival is visible even in the legends that genetics failed to confirm. The Pashtun stories of Greek or Israelite ancestry reveal how communities use genealogy to explain themselves. Science does not need to mock those traditions to test them. It simply asks a narrower question. Does the DNA show evidence that a large part of the population descends from the claimed source? So far, the strongest genetic evidence points instead to deep regional continuity and mixture across South Central and West Eurasia. The legends remain part of cultural history, but they are not replacements for genomic evidence. The Hazara case shows the opposite situation. Here, a historical memory of Eastern ancestry broadly agrees with the genome. Yet even this confirmation is incomplete if it becomes the Hazaras or Mongols. They are not a preserved medieval army. They are a modern population formed from Eastern migrants mixing with older inhabitants of the region and then developing their own history over centuries. DNA supports part of the tradition while also revealing everything the simple tradition leaves out. That is what makes Afghanistan such an important place for population genetics. It demonstrates that human identity is built through layers rather than clean replacements. One layer may come from Neolithic farmers, another from Bronze Age steppe pastoralists, another from local South Central Asian populations, another from medieval Eastern Eurasian migrants. Each layer can be stronger in one community and weaker in another. Over time, endogamy, geography, language, and politics turn those different mixtures into recognizable populations. So why choose roughly 4,000 years for the title rather than 1,000? Because the decisive genetic structuring of Afghan populations did not begin with the medieval empires, evidence points much deeper into the Bronze Age, when steppe movements, oasis civilizations, and early Indo-Iranian population transformations were reshaping the entire region. The Mongol period is crucial for understanding Hazaras and parts of Central Asian ancestry, but it is only one chapter. The Afghan genetic mystery is at least a 4,000-year story of population structure, and its deeper foundation reaches much further back. The next breakthrough may come from a cave, a cemetery, or an ancient city already known to archaeologists. A single well-preserved skeleton from the right period could directly test models that currently depend on neighboring regions. A cemetery, spanning several centuries, could show whether steppe ancestry arrived suddenly or gradually. Medieval genomes could reveal how much Eastern ancestry entered during Mongol rule. Ancient Bactrian DNA could clarify continuity between Bronze Age oasis populations and later communities. Afghanistan has supplied the geography for these migrations for millennia. What science still lacks is enough ancient DNA from the people who actually lived through them. Until those genomes are recovered, the answer is both clearer and more complicated than the old legends. Afghans were never the descendants of one vanished civilization. They were never simply Persians pushed east, Indians pushed north, Greeks left behind, Israelites lost in the mountains, or Mongols who settled after conquest. Different Afghan populations carry different pieces of those wider Eurasian worlds, but the strongest pattern is continuity mixed with repeated contact. The Hindu Kush did not create one isolated people. It created a meeting ground where populations could remain distinct while still exchanging ancestry across thousands of years. And that may be why Afghan DNA looked mysterious for so long. Researchers were searching for origins as though every ethnic group had to begin at one point on a map. Afghanistan does not work that way. Its history was built at the center of roots rather than at the end of one migration. The genetic record is therefore not a straight line leading backward to one homeland. It is a network connecting Iran, the steppe, Central Asia, and the Indian subcontinent, with different Afghan communities preserving different sections of that network. 4,000 years of migrations did not erase the people of Afghanistan. They created the genetic landscape that exists today. The final answer is not one ancestor. It is the crossroads itself, still visible.